Good evening, everybody. My name is Dolores O'Riordan. I'm the Director of UCD's Institute for Food and Health. I'm delighted that you can join us here this evening for the UCD's Institute of Food and Health public lecture. So this lecture series is designed to give the public the scientific knowledge in areas related to nutrition and health, where often there may be some confusion or an area of high interest for the public. So it can be quite daunting in looking at social media, in looking at popular press, to really decipher what is the truth of the matter behind some food and health issues. Tonight, we're going to talk about taste, a very important topic, critical to all our enjoyment of food. We have two speakers, Dr. Emma Feeney and Dr. Claire Reynolds. They're both going to speak to the topic, and at the end of their talk, we'll have a question and answer session. If you have a question and answer, you can just place your questions in the tab, and the questions that you posed will be moderated by my colleague, Professor Lorraine Brennan. We'll try and get through as many as the questions that you can, and for you to share in Emma and Claire's knowledge in the area. I'll hand you over to Lorraine now, and she'll introduce the speakers, and then we'll get started with the talks. Over to you, Lorraine.
Thank you very much, Dolores. Good evening, everybody, and I'm delighted that our two speakers accepted to speak tonight on this very interesting topic. So Emma is going to start off. So Emma is an assistant professor in School of Agriculture and Food Science in UCD, where she coordinates a number of modules on food ingredients, sensory science, and human eating behaviour. Emma has experience in nutrition and sensory science, and her research areas of interest include food consumption patterns, dairy foods and their contribution to health, food structures and the impact on digestion, absorption and assimilation of nutrients. She also has research interests in sensory evaluation and individual variation in taste and flavour perception. When Emma is finished, she'll hand over to Claire, and Claire is an assistant professor in the School of Public Health, Physiotherapy and Sports Science here in UCD as well. Her research interests include metabolic and reproductive health, with a focus on how diet influences the development of obesity and metabolic dysfunction across the life course. Her current research examines the role of artificial sweeteners on male fertility, pregnancy and developmental programming of obesity in the offspring. So again, I'm very delighted that both Emma and Claire took the time tonight to share their knowledge on taste. So I'll hand over to Emma, who's going to start.
Thanks very much, Lorraine, for the lovely introduction. So I'm just going to begin by giving you a quick overview of what we're going to talk about this evening. Whoops. I suppose just to give you a flavour, pardon the bad pun, of what we're going to talk about. We're first of all going to give you an introduction to taste, and really what we mean when we talk about taste is flavour, and hopefully help you to understand that it is really a key driver of food intake. So we'll talk about the difference between flavour and taste, and then we're going to talk about some differences in receptors that drive some of those differences in flavour and taste as well. We'll talk a little bit about taste preferences for sweetness and bitterness, and how those preferences actually develop. And then we'll talk a little bit about those taste receptors and how we actually find them in other areas in the body and where they may play a role beyond taste. Then Claire is going to talk a little bit about the use of sweeteners in the diet, as a mode of energy reduction, but also some other, I suppose, things that may be going on there as well. And she's going to present some really interesting results from some of her recent research on sweetener use. She'll talk a little bit about sweetener use in pregnancy and some dietary intakes as well. So I suppose, why are we here this evening? We really want to tell you a little bit about taste, and food choices is really complicated. So this is, I suppose, just an overview of some of the various areas that are involved in food choice. So you can see that we can really, divide these into various groups. So we can look at some of the economic determinants of food choice, and obviously these include things like price and availability of money. Then we can see that we can also talk about the psychological aspects of food choice as well, and these include things like mood and stress, and then we can talk about the attitudes and beliefs and knowledge about food as well, and these have a really important role in how we determine food choices as well. There are social aspects in there as well. So things around perhaps a social group, or even pressure and peer pressure as well can influence our food choices. We also have the physical aspects, such as physical access to food as well, that can impact our food choice. And then also we have biological determinants as well. So these include things like appetite, satiety, and so on, but particularly taste, as you will hopefully see. So it's really a key driver in food choice. And when we talk about taste, really what we mean is flavour, because it's very much a multi-sensory perception. And I suppose anyone, we talked about this earlier, just before everyone joined this evening, but anyone who has been unfortunate enough to have suffered from smell loss during COVID will certainly know the importance of smell or olfaction to the perception of flavour as well. So flavour really is this multi-sensory perception that comes from taste. So those 5 tastes that we can detect in the mouth, which are sweet, sour, salt, bitter, and umami, which most of you will probably be familiar with, but that's that savoury taste that's really well exemplified by sort of MSG. And then we have a smell or olfaction as well. And we actually have 2 types of smell. So we have that smell or olfaction of the food through the front of our nose, which is known as orthonasal olfaction. And we have the smell retronasal olfaction, which is the smell of that food up the back of our nose when that food is in our mouth. And that's really one of the key, I suppose, components of flavour that particularly anybody who did suffer with smell loss from COVID would be familiar with, because a lot of that retronasal olfaction, where a lot of those volatile aromas that contribute to the flavour would have been lost. And then finally, we have something called trigeminal sensation as well. And this might be a little bit less familiar, but trigeminal sensation is something that's activated through the touch system. So it's also known as chemesthesis. And really this is sort of chemical sensations that occur via the touch system. So you can see here, there's a couple of examples of those. So the heat or the burning sensation that we might experience from capsaicin that's found in chili peppers, this would be activated through something called TRPV1 channel. And then we also, we also have the cold or cooling sensation from something like an ice cube, but then also something like a compound called menthol that we find in mint. So anybody that ever would have, got a bit of toothpaste perhaps on your arm or your hand when you've been brushing your teeth, you would know that, while it's in your mouth, it's also, there are also channels for that across the body as well. So that menthol would activate that touch system. And then also the tingling or buzzing sensation that you might experience, for example, from a carbonated beverage, and those are activated through the CA6 channels. So I suppose all of these things then really contribute to the perception of flavour. And if you just take a moment and have a little look at this picture, when I looked for this picture, I googled good delicious meal, and hopefully this is a representation of something that would be delicious for some people at least. But when you look at this, if you have a think about, a really delicious meal that you might have experienced recently, or perhaps something that really sticks out in your memory as being a really delicious meal, and if you think about it, it probably just wasn't the taste or the flavour itself, typically the smell and a taste alone, but also things like the texture of that food, the crunch of it in your mouth, or if it was something like a nice glass of wine, it perhaps was the feel of the glass in your hand. So there are so many, I suppose, different things that contribute to the perception of flavour when we think of a meal like this. My buttons aren't working here for clicking these slides on. Apologies, folks. Okay, so I suppose that was really just to summarise that there are so many different things that are involved in flavour and in flavour perception. But what we're particularly interested in here this evening is taste. And so if we think about individual differences in people, and we're all very different in terms of our hair colour and our eye colour, and this is because we all have slightly different versions of the genes that encode those different things, and it's actually the same for taste as well, and particularly for, as you can see here, sweet, umami and bitter tastes. So these are the segments that are orange and green and red in the pie chart. And we have loads of different variations in the genes that encode these particular tastes. And so these actually can lead to quite stark differences in how we perceive some of the foods and ingredients that lead to these tastes. So for example, what I want to point out here as well, not to go into too much detail in what the receptors look like or anything, but if you look at umami and sweet here, you'll see that there are two receptors, that they're made from two different subunits, and you'll see that they have two genes each that encode those receptors. So there's two genes that encode the umami receptor, and there's two genes that encode the sweet taste receptor. And we can get little variations anywhere in those genes that can lead to differences in how we perceive those tastes. And what's also really interesting, if you look at the bitter segment here, we actually have over 30 genes that encode bitter tastes. So that's really incredible, you know, when you consider the differences there. And it does seem to have been something that has been driven evolutionarily for us to be able to perceive bitter tastes in our local environments. And again, really, it points to the fact that bitterness was very much something that arose to be able to perhaps alert us to potential toxins in our environment. And again, we'll not go into too much detail in this slide, but I suppose just what I want to highlight here is that some of you may be aware, if we look at the gene here for bitterness, you can see that one of the genes that is really, really well known here is the PTC gene, or the TAS2R38 gene. So this is just one of, and it says approximately 30, and that's because the number of genes actually varies from person to person. So we have these deletions, and so some people might have 25 or 26 bitter taste genes, and some people may have as many as 35. So there's a variation in the number of bitter genes that we have as well. But you'll see that PTC, or TAS2R38, that gene comes up time and time again because it's a really well-known one. So we'll just talk a little bit about it now as an example here. So you may have heard of a super taster before, and there was a whole flurry of research in this area a few years ago, and it really, you know, it's one of the key examples of differences in taste perception that most people are aware of. Most of you who are listening this evening may have at some point done a little taste test where you would taste a little strip of paper, and some people would be what's known as taste blind to that, so it might not taste like really anything at all. And then some people actually may be what's called a super taster. And the research in this area actually goes right back to the 1930s, whenever lab protocols were a little bit more loose than what they are now, and someone was working with a new substance. Well, we'll call it PTC for short because it has a long name. But this substance became airborne, it spilled, and the colleague who was working next to the scientist commented on this really bitter taste. And actually the scientist, A.L. Fox, who was working with it, he couldn't detect it. So they decided to go around and do a taste test, and they fed it to a few different people, again in a quite a scary experiment that you wouldn't necessarily do now. But what they found was that actually a lot of people, 6 in 10, were taste blind to this compound. And so this sort of led, you know, this was the historical background to all of this research, because it was really interesting that the compound that was causing the bitterness was chemically quite similar to these things called ITCs that we find in brassica vegetables. So there's been this I suppose, a lot of curiosity in this area to see whether or not these differences in taste for this compound might actually lead to differences in taste preference. But actually, so there's been a lot of work in that area, but actually there are a whole host of other receptors that are involved in smell perception. So we talked there about taste. There's 2 for bitterness, there's 2 for sweet, there's 2 for umami, and there's over 35 for bitter taste. If we look at the variation then in odor receptors, and here I've just shown you a really brief snapshot of 40 receptors that we know what they do, and we have some data for how they impact smell intensity. But there's actually over 400 known odor receptors, and they're activated in different patterns as well. So each of those different patterns of activation can give rise to a different aroma. And so hopefully what you're able to gather from that is that we actually have a really, really wide range of possible perception for smell, but also for flavor overall as well, when you think that, you know, that all of these things together are what contribute to flavor. So there are really huge differences in how we experience flavor, and everyone is unique. So we all live in our own unique little flavor worlds. And so if we think back to this meal then that we talked about at the start, what this really means is the same meal can be experienced really differently for different people. And I suppose what's really interesting about this is that, this is really key in how our taste preferences develop. And so I just want to show you a quick slide here. And I suppose it's an interesting one because just this week there has been a new report that's come out, a European report that has mentioned that Ireland is really, in terms of our obesity levels, we're really starting to become one of the largest countries in Europe. And unfortunately, it's the same for our childhood obesity levels as well. And so if we look at the different factors that are involved in the development of childhood obesity, we can see that there are a whole range of parental factors here. There's a whole range of environmental factors, and I won't go into all of those. But if we look at the child factors, what's really stark here is there's just one, and it's the development of taste preferences or taste likes. So taste preference or taste likes, they're the single most important child factor that's involved in the development of childhood obesity. And we know that sweet and bitterness and the perception of sweetness and bitterness are really key determinants for this. We know this from research that goes back right back to the 1970s. And again, in experiments that we wouldn't necessarily get ethical approval for now, but where they've given, different compounds to newborn children, and they've looked at the reactions. And so we can see here in the middle, there's a very relaxed orofacial response to sweetness in these newborn babies, whereas over on the far right-hand side, you can see that there's quite a different response to bitterness. And again, a little example from one of my own kids, we recently gave him some spinach. And again, you can see that we do get this innate rejection response for things that are bitter. So taste, and I suppose particularly bitterness and sweetness are key. And again, from some of our own research in UCD, we conducted a large study in over 500 children, and we looked at a whole range of different things. And one of the things that we found was, although there were no major differences in the overall vegetable intakes, we did find that super taster children were 50% less likely to have tried green vegetables. So there is, I suppose, a link there with food neophobia. And again, to point out that was just one gene. So there's been this whole, flurry of research around that one gene that I mentioned. But actually we have at least, 30 to 35 different bitter genes. And again, as we've seen, we have this really wide range of taste and odor receptors as well. So I suppose it is just important to put that into perspective too. And I'm just looking at the time here, and I'm probably going over a little bit, but I suppose just what I want to mention here with this slide is there actually hasn't been a huge amount of research looking at taste and weight. One study in 2012 in a German group of children looked at what they termed a total taste perception score in a control group and then an obese group. And what they found was that the obese group were significantly lower in their perception of taste. And we did something similar in this large study that I talked about in Irish children. But actually what we found, which was really interesting, so you can see the normal weight is in the light blue there, and the overweight and obese group are in the dark blue. And we saw a difference, but we only saw it in boys. So we saw a sex difference where there was no difference in girls for their overall taste perception in terms of their weight status, but in the boys, the overweight and obese boys were significantly more blunted in terms of their perception. And actually, this has been echoed in a number of different studies now, both in Europe and in the US as well, that those sex differences. And again, this diagram is just showing you that really that it's the sweet and the bitter taste differences that are driving those differences. So something really, I suppose, important about the sweet and bitter tastes. And we can see that, this really, these differences in perception and these oh, sorry, I'm just checking the chat there. So these differences in perception and these systems, they start to develop as early as the first trimester. And by the third trimester, they're already fully functional and developed. So that's really, as early as the third trimester before a child is born, we start to get those flavor molecules start to move across from the maternal diet. So maternal diet is really key. And then this diagram, or this slide, is just showing you that actually there is a really key developmental period, both in the neonate and then in the weaning period as well. So in infants, we see again this innate preference for sweet and umami, and this does seem to be very much reflective of human breast milk, which would be rich in lactose, where the sweetness would come from, and then amino acids that would evoke those umami tastes. And again, you know, breast milk, there are, I suppose, key taste and flavour compounds that are transferred in breast milk, and this really provides an opportunity for preference development, because we've got this flavour bridge, as it's termed, from that in utero through to weaning, with that flavour exposure that can happen. And actually, we know this from studies that have looked at some certain types of formula, where we see that some formulas that are extensively hydrolyzed and they can be quite bitter, we actually see a preference in those children for bitterness later on. So there are opportunities there to modulate taste preferences quite early on as well, and introduce those bitter tastes early on as well, and try, I suppose, in some way to modulate the development of those preferences. And then what's really interesting is that actually taste receptors have a role beyond taste. So this diagram here on the right hand side, you can see that the bitter taste receptor genes are shown in dark pink, and then you can see that the sweet taste receptor genes, they're shown in blue. And this is really just showing various areas in the body where they're expressed. So they're expressed in loads of areas in the body outside of the oral cavity, and they seem to have quite specific functional roles. And you're going to hear a little bit more from Claire now in a moment about some of these roles, particularly for the sweet taste receptor genes. But for the bitter taste receptor genes, that's that massive family of genes that are hugely different from person to person. So we see these expressed in the lungs and in the nasal tissue, and they're being used now as targets for asthma therapies. We also see bitter variants are actually linked to reduced risk of COVID-19 in some cases. So where they're being expressed in the nasal tissue and in the lungs, for some people, so again, going back to the TAS2R38 gene that we talked about, there's actually a few studies that have come out this year that have shown that some people who have the functional variant of that gene, so the bitter variant, they have a reduced risk of COVID-19 versus the people who had the non-functional version. And it seems as if, again, that functional version had a role in detecting perhaps the virus and initiating an immune response. And they've also then been found in other areas as well in the body, and where they're used as targets potentially for obesity as well. And I won't go into this in a huge amount because Claire is going to talk about it a little bit now in a moment. But I suppose just to show as well that they have taste receptors, especially bitter receptors, have been found in other areas in the gut, where they seem to be involved in the regulation of food intake. They also initiate a whole cascade of hormones that help to stimulate gut motility after eating. And again, they are promising targets for obesity therapies as well. So I suppose just to summarize all of that, we've seen that flavour is a multisensory perception, and we have a really wide range of genetic variation in taste. There's a link between taste perception and weight. And again, we've seen that some of that may be due to genetic variations in taste receptors, but it's also, you know, there are taste receptors that are found outside of the oral cavity as well that also have a role, not only in the, I suppose, sensing of food and nutrient molecules in the oral cavity, but also in our metabolic response to foods and nutrients as well. So I'm just going to pass you over now to Claire, who's going to talk a little bit more about sweet taste receptors and some of their activations.
Perfect. Thanks a million, Emma. And thanks for that really interesting talk, because it was great. And it has introduced some of the concepts, I suppose, that I'm going to talk about now. So I'll talk a little bit about those taste receptors that Emma has mentioned. Sorry, just start the slideshow. That Emma has mentioned, and kind of how they're activated, some of the endocrine regulation that they undergo. Then I'll go into artificial sweeteners. So artificial sweeteners is something that I've kind of incorporated into my research over the last couple of years, specifically in terms of reproductive function. So we'll have a talk about that. And so I'll show you some of the results that we've gotten over the last couple of years in terms of taste receptors and artificial sweetener function in the testes, but also during pregnancy and beyond. Okay, so I showed you this already, but I'll just highlight here the fact that the sweet taste receptor is very conserved. There's 2 genes involved, the TAS1 or 2 and the TAS1 or 1. These are relatively conserved across species, so you find them fruit flies to whales to humans. They're found throughout nature. And just as an interesting factoid, there are only kind of a few species that don't actually perceive sweet taste, and these include cats and vampire bats. And they think that the reason that they don't perceive sweet taste is just it's an evolutionary feature based on the kind of meaty nature of their diets. And it's not even that the sweet taste receptor is not there. It's just that there's a mutation in it that renders it non-functional. So this gene, because it's so conserved, indicates that it actually has a really important function just in general kind of homeostasis, body homeostasis. So we think of taste perception, you know, in terms of the tongue. You know, you eat your food and you get that taste sensation telling you whether or not it tastes nice, it doesn't taste nice. But actually it's much more complex than that. As Emma had mentioned, you get taste receptors, both sweet and bitter, throughout the body. But they're not as we would traditionally kind of perceive taste. They don't have that link to the brain. And because they don't have that link to the brain, they're not kind of sweet perception per se, but they do have a really, really important role in terms of chemoreception around the body. And this can be really important for a range of biological functions. You know, we see listed here that they're found throughout the body, but we actually don't know a lot about what they specifically do in all of these regions. But we do know a good bit about certain aspects, particularly in the gut, and we're starting to learn a bit more about the reproductive system. So I'll talk a little bit about that in the next couple of slides. So here's what the sweet taste receptor looks like, and you'd be shocked now, but the things that activate it are things that are traditionally sweet, so sugars. So simple carbohydrates like glucose, fructose, sucrose, all that kind of stuff activates the sweet taste receptor. They're also activated by a group of chemicals called artificial sweeteners, which are essentially artificially chemical sweetening agents that can activate these receptors. So what happens once they're activated? Well, they actually have a really, really important role in endocrine function. And if we talk about it in terms of the gut, we actually have a range of sweet taste receptors in our upper gut that perceive the sweet, or that have the sweet taste. And interestingly, patients who undergo gastric bypass surgery can have differences in their sweet taste perception. This may be due to the removal of this region where the taste receptors lie. But what they do is once they're activated, they increase a range of hormones in the gut. And as Emma mentioned, they're related to gut motility, so things like GLP, PYY, and ghrelin. So in terms of gut motility, they also are involved in appetite regulation and other metabolic functions. So they're really important both in terms of metabolic and appetite control. So you can see where the link to obesity might lie in terms of dysregulation of these particular receptors. Another feature of these receptors is they increase the levels of a protein called SGLT. This is a glucose transport protein. So it basically shuttles glucose across the gut and into the system. So potentially overactivation of these receptors could result in an increase in glucose flux across the gut into the bloodstream, which may result in hyperglycemia. Again, something that's highly associated with obesity and metabolic dysfunction. So if you take a look outside the gut, we also see these taste receptors in the pancreas. Pancreas is obviously a really important metabolic organ. It produces insulin. And we see that activation of these taste receptors can increase insulin concentrations in the system. So it clearly has very important roles in metabolic health. So in addition to activating a range of endocrine factors, we also see that these receptors are themselves regulated by other hormones. And a really nice example of this is the hormone leptin. So leptin is produced by your fat cells. So it's increased with obesity. It's involved in appetite regulation. So a really nice study that was carried out in Japan a couple of years ago looked at normal weight individuals. They monitored them over the course of 24 hours. They took blood and did taste preference tests. And what they saw was as leptin went up, taste receptors went down. So there's a really nice inverse correlation between leptin expression and taste receptor expression. They also found that there was a circadian cycle to this. So from noon to midnight, you see this upregulation of leptin and then a decrease in sweet taste perception in the same period. So again, this kind of points to a role in kind of metabolic kind of obesity related factors. So potentially this receptor could form a nice therapeutic kind of or drug target for the treatment of obesity going forward. Once we learn a bit more, we might be able to kind of decipher that out a bit more. So the same group, they carried out the study a couple of years later, the exact same study over 24 hours in a group of people living with obesity. And they found that they didn't see the same level of regulation between leptin and the sweet taste receptor. So again, it all points to kind of a really critical role in these metabolic processes. So one thing that we don't know a lot about is the role of taste receptors in reproductive systems. We know they're there, but we don't necessarily know exactly why they're there. So we were interested in looking at artificial sweeteners within the context of reproductive function. So that's what I'll focus kind of the rest of my talk on. And just for anyone, I'm sure you all know what artificial sweeteners are. They're chemicals that have that activate these taste receptors, but they can be up to from 200 to 20,000 times the sweetness of sugar. So they're highly sweet. They're really, really prevalent in the food chain. So Breach McNulty's group here in UCD, they work a lot with the NANS study, and they've shown that, that a lot of these sweeteners are very prevalent in Irish populations, from childhood populations right through to adulthood. So it's something that I'm sure you've all encountered along the way somewhere, whether you know it or not. Artificial sweeteners, we know they're in diet drinks and things like that, but they can be hidden in other places as well. We see them in kids' medications, so things like Calpol and things like that, effervescent vitamin tablets, chewing gum, toothpaste, whole range of things contain artificial sweeteners. So they're very, very prevalent in our food chain. And I suppose there's a kind of a question over whether or not they're good for you or not. Are they the healthier option in comparison to sugar? You know, some people perceive them to be, but it might not necessarily be the case. And what we see is an association between artificial sweetener consumption and a range of health complaints, such as weight gain, type 2 diabetes, stroke and heart disease, and dysregulated gut microbiome, which again can be an influencer for a range of health outcomes. So this just highlights that artificial sweeteners might not be the panacea to kind of replace sugar. And, you know, we might need to consider kind of their role in the food chain. So that brings me on to my favorite subject, reproduction. So there was a really, really nice study a couple of years ago that kind of consolidated all the data in terms of sperm counts in Western society. So they looked at a whole range of countries, 20 to 30 countries over several decades. And what they saw was a 50% decline in sperm counts in men over this time period. Now, we're not talking about Handmaid's Tale level dystopian futures or anything like that, but it is certainly a worrying trend that we're seeing this dramatic reduction in sperm counts. And the author speculated a variety of reasons could be the source of this kind of this reduction, from sedentary lifestyles, environmental pollutants, to advancing age of the population. But I suppose the one that I'm most interested in is diet. And they've shown, you know, there's plenty of studies out there showing that increased BMI, poor diet, that are rich in saturated fat and high in sugar can have an impact on sperm outcomes. I suppose when we're thinking about sperm and reproduction, we all know the sperm have a job to do. They have to get from A to B within the female reproductive tract. It's not a random process. It's actually a very, very tightly regulated process. So they need to have cues to know how to know where they're going, basically, so they can kind of get to their endpoint. They do this through chemoreception. So sperm have chemosensory abilities that guide them through the reproductive tract so that they can reach the egg successfully. So it's not really that surprising that they contain both taste and olfactory receptors that allow them to sense this environment and make their way to their final destination. And so there is a little bit of work done in this area. There's studies in mice that don't contain the sweet taste receptor, and shows that they're infertile. And the reason that they're infertile is because their sperm just don't mature. When you look in the testes of these animals, there's massive dysregulation and disorganization within the testes themselves. And this results in the sperm just not maturing. So they can't fertilize an egg or whatever. They're also thought to be involved, the sweet taste receptors are thought to be involved in, and also bitter taste receptors, but I'll just talk about the sweet taste receptors for the purpose of this talk. They're thought to be involved in this chemosensory kind of transition through the uterine tract, as well as they're involved in the swimming behavior of the sperm. And sure, if the sperm aren't motile, they're not really going to go anywhere. So with all this in mind, and this clear kind of link between the sweet taste receptor and fertility, we wanted to have a look and see if artificial sweeteners had any impact. So a really talented PhD student, Michelle Kearns, and her first task when she started in the lab was to do a systematic review in this area, look at the association between artificial sweeteners and male reproduction. She didn't find any papers in human studies, and she did find a few in terms of rodent studies. She only found 9 papers in all, so there's actually a shocking lack of evidence around the role of sweeteners in male fertility, given the prevalence of artificial sweeteners in our population. And so it was hard to kind of tease anything solid out of it, because these artificial sweeteners were given in such high doses. They were essentially toxicology studies rather than functional studies. So we set about trying to kind of fill that gap in the literature, I suppose. So we took a group of mice, we had 4 groups, and we fed them a range of different sweeteners. So there was one group, we just gave them water. There was one group, we gave them a sweetener, an artificial sweetener, sulfame K. We had a group that had the bioactive component of stevia, which is riboside A, and we had a caloric sweetener in fructose. So as I mentioned before, a lot of the studies that are done in this area, they have these massively high doses of the sweeteners. They're just not realistic. You'd never be able to translate these findings to a human situation. So we wanted to make sure within the context of our study that we set the dose at a point that could be translated. So the dose that we gave them was essentially equivalent to a human having a can of fizzy drink a day, whether it was diet or caloric sweeteners. We put them on these diet for an 8-week period, and this was enough so that this process of spermatogenesis or production of sperm could go through at least one cycle. After the 8 weeks, we took sperm and we looked at the functional abilities of these sperm. Before we did that, though, we looked at the testes. We wanted to see if our taste receptors were expressed in the testes, and lo and behold, in response to both ACE-K, the artificial sweetener, and stevia, the natural sweetener, we found that there was a significant upregulation of this taste receptor in the testes of these animals. So how did that relate to sperm function? Well, we looked at sperm viability. So were they dead? Were they alive? And there wasn't really an effect with stevia, but we did see a dramatic decline in sulfame K and fructose fed animals. So it's clearly having some sort of effect on these sperm. We also looked at abnormalities in the sperm. So if sperm have abnormalities, this can be kind of, bits missing or bits that are not functional. So kinked tails or bent heads, things like that. We characterized them and we looked at the percentage that were abnormal versus normal. And we saw that there was a significant increase across all of the sweeteners in the amount of abnormal sperm that we had in our samples. So clearly there's something going on here between artificial sweeteners and sperm morphology and viability. We also looked at testosterone. Obviously, it's the main kind of androgenic hormone in males, and we didn't really see any major effect in terms of our testosterone concentrations there. So this is all very good, but we need to further this work, and this is currently what Michelle's PhD is based on. So our next step here is we're going to look and see if these mice can even mate. Can they produce offspring successfully? And if they can produce successfully produce offspring, how do these offspring fare? Have they got any sort of complications? And we'll monitor them over time and see how we get on. So watch this space. We'll have another public lecture in a year or so to fill you in. So that brings me to females. So taste receptors, are they involved in pregnancy? Well, yes and no. So a really nice example is the fruit fly. When fruit flies have eggs on board, they have this really increased attraction to polyamines. So polyamines are a chemical that are produced in fruit that are kind of going off. So they're overripe or kind of on the verge of being rotten. Why is this important? Well, these polyamines are really important for reproduction, and they have a role in kind of reproductive success in these flies. They produce more offspring, their offspring are more robust. So this kind of taste, kind of linked with taste and pregnancy, is there, at least at a kind of fruit fly level. But there's obviously a very big jump between fruit flies and mammals. So we had a little look and saw that indeed, in mice at least, during pregnancy, there's a decrease in sweet taste reception, and this is associated with a reduction in the sweet taste receptor. The authors speculate that it's a potential mechanism for increased caloric consumption during pregnancy. And I suppose this is probably obvious enough. You know, women go through an enormous number of physiological adaptations during pregnancy, and taste can be one of them. We all know about pregnancy cravings. So we had a look in the literature just to see if there was any association there. And actually there is, but it's really clouded. There's no real consensus on whether or not pregnancy can influence taste perception in human females. And some studies say they can, some say they don't, and there's anywhere in between. And this may be a feature of the study design. You know, they've taken, you know, all the studies take their information at different points in pregnancy, and you know, the hormonal milieu across pregnancy can change quite dramatically between trimesters. But we do know that some of these hormones, so leptin obviously is increased during pregnancy, and things like estrogen and progesterone, and these all have the capability to affect taste receptor function. So it stands to reason that there is going to be some sort of role for taste receptors during pregnancy. But, you know, the evidence in terms of humans just isn't really there in a consolidated form just yet. But we were interested in how artificial sweeteners fare. And actually there's a little bit of data kind of emerging in some of the birth cohort studies around the world. So there's some really nice studies, kind of observational studies, that have associated artificial sweeteners with things like increased glucose tolerance or intolerance during pregnancy, increased risk of preterm birth, and increased risk of offspring obesity up to the age of 7 years old, where the children have slightly increased BMI compared to their peers. And it's not surprising, because artificial sweeteners actually do get through, well, some, I'd say a few of them don't, but a good few of them get through into the amniotic fluid. And as Emma mentioned, breastfeeding is really important for setting those early life cues for taste preference. And we know that 65% of breast milk samples have some sort of artificial sweetener in them. So it gets through into the milk. So presumably it can be, you know, you can get exposure via that route. So certainly artificial sweeteners, there is some observational evidence there to show that they do have an impact during pregnancy and on the next generation. So again, we wanted to tease this out a bit further in our preclinical models. So we set up this study. So there's a few different arms to it. The first one is to look at pregnancy, see if artificial sweetener or our caloric sweetener fructose could have an effect during pregnancy in terms of their metabolic function. So we looked at oral glucose tolerance tests. So any of you who were ever pregnant and had to have a GTT, you'll know how the poor mice felt when they had that procedure. We also looked at the offspring of these animals. So we fed the mothers the artificial sweetener and the fructose during pregnancy and lactation, and the offspring were maintained in just a normal low-fat diet for the rest of their lives. We also had a third arm where we monitored the mothers after weaning. So once the mothers weaned, we separated them and we just kept them on it again, a low-fat diet for 6 weeks post-weaning. So they weren't exposed to the artificial sweeteners during this period. So that was kind of the general kind of layout of the studies. And again, we used these moderate doses that equated to about a can of fizzy drink a day. So at least we hope these could be translatable to a human situation. So what we see when we look at our oral glucose tolerance test, and for those of you who are not familiar with this, you basically give a drink that has a set amount of glucose and you monitor blood glucose over a period of 2 hours. What we see is elevated glucose concentrations in our artificial sweetener animals and our fructose animals. And this indicates that they are metabolically compromised in some way. So they don't deal as well with fructose as, say, a control animal would. So there's definitely issues there. When we looked in the guts of these animals, as I mentioned before, we have these sweet taste receptors in the guts. We see that TAS1 or 2, which is a component of the sweet taste receptor, is significantly elevated in our artificial sweetener animals. So there may be a link there between the sweet taste receptor and these kind of elevated glucose concentrations. We also looked at some more pregnancy reproductive type parameters, and we saw that of the animals we mate, significantly less mated in the groups that were receiving the artificial sweetener and the fructose. So these were significantly less likely to get pregnant. And of those that did get pregnant, their pregnancies were shorter. And now it's hard to link preterm birth to a rodent model. They don't really, but it is interesting that we do see this sort of parallel between the human situation and the animal situation, with the shorter pregnancy length. We also looked at fetal glucose concentrations, and the reason we looked at this is often women who have metabolic complications during pregnancy, things like gestational diabetes, their newborns often have hypoglycemia and have to be monitored for a few days. And similarly to this kind of situation, we see that elevated glucose in our mothers, and then with our fetuses, we see a reduction of hypoglycemia in our fetuses in the artificial sweetener and the fructose groups. So there are some nice parallels between this study and human situations. Obviously, they're mice, they're not humans, so there's only so much you can take from these kind of studies, but it is interesting that we do see these kind of parallels. So as I mentioned, we did take some of the mothers on a bit further. We looked at them at 6 weeks post-weaning. When we did our oral glucose tolerance test to look at their metabolic control, we actually saw that even though they hadn't received the artificial sweetener or the fructose for at least 5 weeks, 5-6 weeks at this point, they were still metabolically compromised and they still had elevated glucose during their GTT. This was in the absence of any differences in body weight, fasting insulin, fasting leptin, and fat mass. So it is interesting that we're still seeing these kind of effects. And it does need to be teased out a bit more to see what the mechanism behind all this really is. Another interesting thing that we found in these mothers was when we looked at their ovarian function, we saw that they had elevation in genes that were involved in a process called folliculogenesis. And this is key for estrous cyclicity and ovulation. So potentially these animals may have alterations in their reproductive function. It's something that we need to follow up on, but it is interesting that we are seeing these changes in the ovaries of these animals. So if we go on to their daughters, we monitor these animals for 16 weeks. So I guess this would be the equivalent of young adulthood in humans. And when we looked at their ovarian gene expression, again, we saw this dysregulation in genes involved in folliculogenesis, indicating some potential reproductive dysfunction. So we monitor their estrous cycles for 5 weeks. So the estrous cycle of a mouse is 5 days. So it meant that we could have a significant number of cycles to make sure that our results were reliable. So we had at least 5 or 6 cycles from each mouse to just determine whether or not what we were seeing was real. And what we saw was that in mice whose mothers were receiving artificial sweetener and fructose, so these animals were exposed in utero to these compounds, we saw a significant increase in estrous cycle dysregulation. So they had a bit of a reproductive profile here. So we also looked at testosterone, and the reason we looked at testosterone is I'm sure some of you are familiar with the condition polycystic ovarian syndrome, and a feature of this is an increase in testosterone that creates metabolic and reproductive dysfunction. And similar to this kind of condition, we saw an increase in testosterone in the offspring of mothers who've been fed artificial sweetener and fructose. So again, we're seeing this kind of reproductive metabolic dysfunction in these animals. So just to finish up, this is all an artificial sweetener, but if you look at high-fat diet exposure during pregnancy, we also see some dysregulation in terms of taste. So what we see is when animals are exposed in utero to high-fat diet, they have increase in the sweet taste receptor, and this applies to both males and females, although females do seem to have a more potent effect. When you go on to test their taste preference, we see that during pregnancy, or animals who are exposed to high-fat diet during pregnancy, but not lactation, have an increased preference for sweet taste as adults. So there's definitely some association here between early life factors and preference for taste. Just to highlight, these are rodent systems, but they can be quite useful in teasing apart the mechanisms that you can't really do in humans because of the complex feeding behaviors that Emma would have discussed earlier on. So just to sum up, we see that taste is a key driver of food choice. There's a wide variety of taste and smell that impacts flavour. Sweet and bitter sweet and bitter tastes are key determinants for taste preference. We have key roles in multiple organ systems for these taste receptors. Potentially, they could have a therapeutic role. And artificial sweeteners, they play a role in fertility, reproduction, and there may be intergenerational effects. Thank you for listening, and thank you for any questions that you may have. We're more than happy to answer them.